All right, let's wrap up this uh, brief introduction to the control of gene expression. And it is a brief introduction, and it is fascinating. And if you have questions or if you want to dig a little deeper about anything that we've talked about or what we're going to cover today, please reach out to me and let me know, and I'll do the best I can to guide you and to help you uh, learn and understand anything you are interested in. So, in the first couple of videos, we've talked about general ways that organisms control the expression of their genes, and that means control whether or not the products of the genes are going to be made in a cell and how much of that product might be made in a cell. And by controlling this expression of genes, which usually means making a protein or not, and how much of the protein, controlling the expression of genes in this way is the way organisms control the development of specialized cells and tissues and organs, and it's the way organisms adjust to their environment and to the changing conditions that they're exposed to so that they can survive in a very dynamic and ever-changing world. We're going to take a look at an example now, because sometimes an example is the best way to illustrate a point. And I opened up the first video in this series talking about why do liver cells act like liver cells, and why do the cells in the lens of the eye act like lens cells and not liver cells. And here's a closer look at that phenomenon. So inside a liver cell, there are all the genes that a human cell would need to do the work of any human cell. And inside a lens cell, the lens of our eye, inside the lens cell, likewise, there are all the genes for controlling the functions of all the different types of cells in the human body. So what's going on to cause the liver cell to be so different from a lens cell? Well, liver cells, for example, make a lot of a particular protein called albumin. It's a common protein made in vertebrate animals. And they don't make any of the protein crystallin, which is a protein that cells in the lens of the eye make. And those crystalline proteins build the transparent structure of the lens so that light can pass through and be focused onto the retina of our eye. So lens cells need to be able to make crystalline so that they can make a lens and keep a lens healthy and functioning. They don't need to make albumin, a protein that's necessary for the functioning of liver cells. Liver cells need to make lots of albumin, and they don't need to make any crystalline. So what's going on? Well, as the liver cell develops, as the organism, in this case a human, develops, and the liver starts to develop inside the embryo and then inside the fetus, genes are switched on that produce a whole series of transcription factors. Lots and lots of activators and lots of repressors too. In the liver cell, activators are produced that bind to DNA and switch on the gene for making albumin. And so the liver cell starts to act like a liver cell. And at the same time, that liver cell would make inhibitors that would bind to DNA at sites around the crystalline gene. Liver cell does not want to transcribe any of those genes, does not want to express them. And so these repressor proteins will bind to control regions around the crystalline gene and switch it off. And in fact, probably just lock that sucker up for good. liver cells are probably never going to make any crystalline. So just go ahead and lock up that section of the chromosome. We're never going to have to transcribe those genes. In the lens cell, as it develops, because of the signals it gets, it switches on genes that produce transcription factors that do go to the site near the crystalline gene, and they bind to the DNA, and they enhance transcription. And so RNA polymerase comes along and chugs through the DNA, transcribing the messenger RNA, and the lens cell makes crystalline. And likewise, to the liver cell, those proteins that the lens cell does not need to make, those are going to be controlled by transcription factors that bind to control regions and deactivate that gene, that repress the transcription of that gene. And so as those two different tissues, two different organs develop in our body, a whole parade of different transcription factors are made, and certain genes are activated or are enabled and maybe activated later. Other genes are repressed and in some cases locked up for good because they will never need to be expressed in that cell. One last thing, and then we're all done. And this is a really interesting branch of developmental biology, and it's the expression of groups of genes that are called homeoboxes, and that's because of their structure. And if you get interested in learning more about this, we can go into why homeobox is the name. And they go by the abbreviation HOX. So homeobox genes, or HOX or HOX genes, are ones that code for transcription factors that control the differentiation of cells and tissues and embryos, just like these transcription factors we talked about in the liver cell and the lens cell. So as those cells and those tissues that they belong to, as they're developing, genes like these homeobox genes are switching on and making the correct transcription factors that will have that cell start to develop in a particular way. The lens cells will start developing and acting like lens cells. Liver cells will start developing and acting like liver cells. Well, the interesting thing about these homeobox genes, these HOX or HOX genes, is that they are arranged on chromosomes in a way that mirrors their effects on the bodies of the organism. And it's more pronounced in simpler organisms like insects and worms. But that arrangement of the genes, the HOX genes on our chromosomes, still retains some of that spatial mapping that is reflected in the bodies of the organisms and how those genes control the development of those particular regions in the embryo as it grows and develops. HOX genes are descended from genes of common ancestors, and I said not surprisingly, they are highly conserved, meaning these genes stay unchanged throughout evolutionary history. And that's not surprising because these control mechanisms that determine how an egg can successfully become a larva or an adult animal, there are a bazillion things that can go wrong, and it's miraculous that any of us are ever born and grow up because of all the things that could go wrong and don't. And so the genes that are expressed that help control this development, the steps that lead to the normal development of a complex multicellular animal, those genes, those control genes, are highly conserved. They have been used throughout evolutionary history to help map out the bodies of multicellular organisms. And here's a confusing, perhaps, diagram, but I hope it will give you an idea. And if you look up here in this simple roundworm, there are certain genes that start to switch on in different regions of the embryo as it develops. Those genes are arranged in the same sort of linear order on the chromosome of C. elegans. And those arrangements line up with the arrangements of the different parts of the animal's body from left to right on the animal's body, from head to tail, and from left to right on the chromosome. Similarly, in insects, highly conserved arrangement from head to tail, these homeobox genes on this chromosome of a fruit fly arranged one after another in the same linear arrangement on the chromosome. This is a primitive chordate animal, a relative of vertebrate animals. And again, same arrangement on a chromosome. And then, for humans, we have our homeobox genes arranged on different chromosomes, but this linear arrangement on the chromosomes is still apparent, and it still controls a sort of head-to-toe development in our embryos and then in our bodies as we are born and grow and develop. Crazy. All right, this is a summary of a lot of the things that we talked about. You don't need to hear me talking anymore, so you can pause the video and read these. And here are some questions for you to think about and, at least in your head, answer to see if you're making sense of some of the big ideas in the control of gene expression. Thanks for sticking with me through a whole bunch of videos. And please bring your questions and ideas to share with me and your classmates next time.